Hello, my name is Camila, and today I will be talking about stem cell culturing, with most specifically pluripotent stem cells. Stem cells are cells that are undifferentiated with no structure or functions. Stem cells have the unique ability to self-renew or differentiate into various cell types in response to appropriate signals. These properties provide stem cells with unique capabilities for tissue repair, replacement, and regeneration, making them valuable research tools in regenerative medicine and therapies. Stem cells can be broadly characterized into multipotent or pluripotent stem cells. Multipotent stem cells can self-renew or differentiate into specialized tissue-specific cell types. Below are some examples of those multipotent stem cells. Then, pluripotent stem cells can differentiate into any cell lineage. They are classified based on the tissue of origin, and there's also 3 types under this one. I have also added a table that looks at stem cells and the selected culture conditions for each. Here in Figure 1, we look at clinical trials for cell therapies using pluripotent stem cells. This data was pulled in September of 2020 from clinicaltrials.gov for all active and recruiting clinical trials involving humans. Next, we look at a guide for passaging human pluripotent stem cells. First, you must prepare your culture substrate by either using a feeder cells or a defined extracellular matrix coating to support the growth. If using frozen pluripotent stem cells, thaw them quickly and gently to maintain cell viability. Then transfer the cells to a pre-warmed tube containing a suitable medium to neutralize the cryoprotectant. Next, begin the seeding process by plating the pluripotent stem cells onto the prepared culture substrate, ensuring the medium is supplemented with essential growth factors such as BFGF. Maintain routine feeding by replacing the medium daily or as needed to provide fresh nutrients and growth factors. Monitor cell density to prevent overconfluency. Once the cell colonies reach an appropriate size, they can be passaged to maintain pluripotency and prevent differentiation. They detach cells using enzymatic methods and collect cell clumps and plate them onto fresh culture substrate. Then monitor by regularly checking cultures under a microscope to assess cell morphology and perform routine quality control assays. If differentiation is desired, modify the culture conditions by removing specific growth factors or adding differentiation-inducing factors. Then freeze the cells as needed for long-term storage using a cryoprotectant solution and store in liquid nitrogen. Additionally, please keep in mind that the protocol may vary depending on the type of pluripotent stem cells used and the specific culture conditions that are best optimized for your cell line. Next, we look at Figure 2 taken from a 2017 Primary Journal article that looked at pluripotent stem cells in neuropsychiatric disorders. Here, we look at schematic representation of induced pluripotent stem cell generation and application. A biopsy is taken from a patient, such as a skin biopsy or a blood biopsy. Then patient cells are reprogrammed into pluripotent stem cells and differentiated into neuronal cell types of interest. This study showed that patient-derived neurons could be used for elucidating disease mechanisms, high-throughput drug screening, drug testing, and toxicity studies, as well as biomarker identification and patient stratification. Pluripotent stem cell culturing have several limitations. First, achieving controlled and efficient differentiation into specific cell types can be challenging. Continuous passaging may lead to genetic and epigenetic alterations, and the use of embryonic stem cells raises ethical concerns. Next, there are also high risk of tissue rejection, as well as potential teratoma formation, which is a germ cell tumor. In a 2010 study by Medvedev, plasmid vector showed low yield. In 10 experiments, the yield varied from 1 to 29 pluripotent stem cell colonies per 10 million fibroblasts, whereas up to 1,000 colonies per 10 millions were obtained in the same study using retroviral constructs. This study also found challenges with the recombinants. Either it had incomplete excision of integrated sequences, or the recombinant did not integrate the allogenic DNA into the host genome. There are also many advantages with pluripotent stem cell culturing. First, it enables the study of basic biology, development, and disease processes. It provides a potential source of cells for repairing or replacing damaged tissues and organs, and it offers a platform for testing the safety and efficacy of drugs. It is also easy to isolate and grow and has unlimited self-renewal opportunities. Here's another figure that looks at the various stem cell types that exist, including totipotent, pluripotent, and multipotent stem cells. When comparing pluripotent stem cells to the other techniques, it is the most similar, with pros and cons, to totipotent stem cells. It is also the least similar to multipotent stem cells. They both have different sources of origin. Pluripotent stem cells begin with blastocytes, umbilical cords, or reprogrammed stem cells, while multipotent stem cells begin through organs with specialized tissues. Multipotent stem cells are also hard to isolate and have limited differentiation, while that's the opposite for pluripotent stem cells. It is also important to note that there are many types of stem cells, and it all depends on your cell lineage that you are trying to start or your end therapy goals. However, pluripotent stem cells is one of my favorites just because of its broad range of sources that you could start with. Next, when looking at controls for pluripotent stem cells, there are negative controls, positive controls, and quality control measures. Negative controls includes cultures without stem cells to ensure that the observed effects are due to the presence of the stem cells and not due to other factors. Positive controls include cultures with known outcomes to validate the efficacy of differentiation protocols, and quality control measures include regular assessments of cell identity, genetic stability, and sterility. For the last figure, we look at human pluripotent stem cells for clinical use. This was taken from a primary journal article, and it looks at the culture media for human pluripotent stem cells contained in animal-derived components that could pose a hurdle for therapeutic use in humans. However, most of the issues that have been solved through recent technology innovations, such as the use of integration-free methods and xeno-free culture. I chose this specific image because it shows what pluripotent stem cells should look like when grown in culture. And lastly, how does this technique tie into our module? We have learned about cell culturing, which the stem cell culturing shows very similarities to what we learned in class. Additionally, newly discovered gene editing technologies like CRISPR, which we also learned in class, may advance stem cell research and offer promise for treating difficult disorders. So not only did we learn basic similarities when culturing those cells, but also other lab techniques that we learned could be used with this process. Here are the references used for this presentation. Please let me know if you have any questions, and I hope you all enjoyed.